Thank you very much. It's a real honor and pleasure to be back here again at Medicine X. And I'm going to spend the next 15 minutes talking to you about big data in biomedicine, one kind of organic data. And I think you've now spent the last 2 days, almost 2.5 days, just learning about a lot of different new technologies in medicine. I think you've heard a lot about omics technologies, genomics, and DNA. And I'm here to talk about this kind of data as well. On the left here, you see what I live and breathe today at Stanford in my research. An example of one of these high-throughput technologies in medicine. This one's called a gene chip. And because data is too virtual, too ephemeral, I have a prop here, which I always bring. And this is what it looks like in real life. And this is a gene chip. And this gene chip now lets us quantitate every gene in the genome. It's kind of designed by design, looks like a computer chip. And that lets us measure every single gene in the genome. And it's kind of amazing. I got my start in this field with the first 60 or so of these chips. We're looking at different cancers, and we used to measure them one by one. On the right here, on the slide, you see when we get tired of measuring them one by one, we measure them 96 at a time. So that's the 96-well format of these chips. So instead of one by one, each well in that plate has a different chip. I've said this before. I think it's kind of funny how computer technology in Silicon Valley scales by the number 1024, right? Kilobyte, megabyte, gigabyte. Life science scales with this number 96. So we have 1, and then we have 96 wells, and then 384 wells. I think the next one's 1536 or something like that. So life science scales at a different number than IT. It's kind of telling. Now, this chip is amazing. This technology is astounding. We can measure every gene in the genome, and it's actually only a couple $100 for each of these samples, plus labor, of course. And on top of that, we now have had these chips for more than a decade. We've had these almost for 15 years. And what happened is the researchers who started to use these chips, they started to write papers about them, publications. And the journals started to realize we're getting too many papers with these chips, these microarrays. And so what did the journals say? They said, no more papers with these microarrays, with these chips, unless you deposit your data into an international repository so other people can get their hands on this data. And these repositories started about 10 years ago, and they've been growing ever since. And something magical happened last year, August 1st. We hit 1 million of those chips publicly available today, 1 million of them. So I started with 60, and you can see the growth chart there. 2000, we're down near 0 on the bottom right. And then you see how we're up to 1 million. About 3/4 of them in the United States repository, another 250,000 in the European res. And it's kind of sad. It looks like it's slowing down there, right? This trend. It's kind of really sad what's happening. And then you realize they only counted half the year last year, right? This came in August. So we're still doubling about every 2 to 3 years, just about finally slowing down to Moore's law. And just to make it clear where these websites live, because people ask me all the time, I'm showing you 2 screenshots here, 2 dumps. The top is the National Center for Biotechnology Information, NCBI. It's part of the National Library of Medicine, which is part of the National Institutes of Health, NIH. And you can see there, this is as of 2 weeks ago, 929,000 samples just there. And the bottom is the European repository run by EBI, European Bioinformatics Institute, and they have another 250,000. So actually today we have 1.25 million chips samples publicly available. 1 million was eons ago, right? We got 1.25 million already today. And some of you have heard me say this before. I'm going to say it to make it obvious here. You saw Jack Andraka giving a keynote, so you know what a high school kid can do. Let me tell you what I think a high school kid can do. On the top of this website there, you see a simple search box. And what I like to say is that even a high school kid now, she needs to do a science fair project. A high school girl can type in the word breast cancer there and hit go. And today, she can go download 39,800 samples of breast cancer. 39,000 samples of breast cancer, about as easily as she could find a song on iTunes today. Now, it's kind of neat. Almost 40,000 samples of breast cancer already digitized, already freely available. It's not just actually humans, so most of them are human samples. On the right there, you see mouse and rat models, even dog models, and even viruses that might be causing breast cancer. All of this publicly available. So 40,000 samples is kind of interesting, but it's easy to gloss over that number. The more interesting number to me is the higher number there, 1,500. There's 1,500 independent labs that have deposited data like this. 1,500 labs have run these experiments and deposited data. So let me tell you how amazing that number is. Let's say I came to you with a new grant proposal or business plan. I come to you, I'm asking you for money, and here's my plan. I'm going to get 1,500 of the best labs in the world to take their best examples and samples and patients with breast cancer, run the latest technologies, and all these folks are on the internet, and they're going to give all that data to me, right? So kids today would call it crowdsourcing, right? So I'm going to crowdsource breast cancer research to 1,500 labs, and I'm not going to pay them a dime to do any of this work. If I pitched this to you in a forward direction, you would laugh me out of the room. Why would these 1,500 labs help you at all today, right? And so what I like to think of as open data, big public open data, to me, is retroactive crowdsourcing. The crowd is helping you, and they don't know it. The crowd is helping you, and you don't know it. But there are the samples, and they're waiting for you to do something with them. And if you're not happy with 40,000 samples, wait another 2 years, there'll be 80,000 samples because it continues to grow even while we're sitting here. What an amazing world it is. Now, if you have any doubts, of course you've seen Jack Andraki giving his talk. If you have any doubts, a high school kid can use this. Here's the one I like to talk about. Brittany Wagner, I've never met her, in Sarasota, Florida. 2 years ago, she won the top prize in the Google Science Fair competition because she used public data, histopath data, as well as clinical data to come up with the artificial brain, artificial neural network for breast cancer. She won the top prize in the Google Science Fair competition. That was 2 years ago. And then this year, she says, oh, let's just do this for leukemia as well. So she gets in the front page of many newspapers doing the same thing a year later for leukemia. So indeed, a high school kid can do this. And when I'm saying a high school kid can do this, then I'm really trying to say is there's no reason you guys can't do this either. Any of you, any of us. That's what the data's for. It's not just for a select few, it's for everyone. That's the point of open data here. Now, in my lab, we tend to collect a lot of this data. In fact, my first big grant at NIH grant here at Stanford was just go get every disease. Why even just stick to breast cancer? Let's just go get all of them. We get them all, and we came up with this big grid like this. We call this a human disease gene expression collection. You got diseases on the rows there going up and down, like leprosy and HIV and, of course, cancers. You got 20,000 genes along the bottom there, different genes. And if you can see the colors, the blues mean that the gene goes down in the disease, and yellow means it goes up. And it doesn't even matter which gene goes which way, but we start to see patterns between these diseases, patterns. So like, this disease and this disease are similar to each other, and we as physicians never knew this. You start to get interesting patterns. And as we're collecting all this disease data, it turns out people, especially folks at the Broad Institute, at MIT, and many others, started to deposit drug data as well. Here are cells treated with the drug, without a drug. Here's a disease sample with drug, without drug, with drug, without drug. So just to match these two, or mash these two up, on the left here, I'm showing you we have patients with disease and without disease, and we come up with a mathematical signature there. And on the right, we have cells with a drug, without a drug. And all of a sudden, you realize we can just put these two together, right? And I'll make the statistics super simple for you with my arms, okay? If I've got a disease where this gene goes up and this gene goes down, and now I got a drug in the other database that makes this gene go down and this gene go up, maybe this drug and disease are made for each other, right? Now, of course, you can't be naive about this. Not every drug goes to every part of the body, so you got to match these things up. You got to know a lot of things about these drugs to learn about them. But in general, we can get a new idea for these drugs. And in fact, where it really starts to get fun is not doing this for a new drug, but to do this for an old drug, a drug that we already use in the clinic, maybe a drug that's already safe to use in practice. If we can find new uses for these drugs, boy, it's a miraculous thing sometimes. So our first big story on this came out about a year, almost 2 years ago, was to test this out for Crohn's disease. You just heard a whole lecture about Crohn's disease. You're hearing about inflammatory bowel disease. And we found an epilepsy drug, a seizure drug, works on Crohn's disease. So these are my famous rat colonoscopies. On the left here, you can see this is what a rat colonoscopy looks like, a normal healthy rat colon. This rat is still alive when we do these experiments. In the middle, you can see what it means for a rat to have Crohn's disease. You can see the redness and a little bit of inflammation as it gets there. And on the right is a rat with the same inflammatory bowel disease like the middle rat, treated with an epilepsy drug. And you can see it's all better. That's kind of exciting. It got us really excited. And we started to do more and more and more of these. And one of these actually just came out last Friday. So I had to send a slide early for these guys, so I had to call this drug X, but I'm going to tell you the name of this drug in a moment here. This is a mouse that has small cell lung cancer. And this is work by Julian Sage and Alejandro Sucadero, as well as Nadine Johnson, who actually works with these mice. They knocked out these three special genes to make this mouse get its own lung cancer. So it doesn't have to smoke like how the rest of us get lung cancer. This one gets it in a couple weeks. And on the right is the same mouse, same kind of mouse, treated with an antidepressant, okay? It's a drug for depression. Now, we don't use this drug for depression anymore because it's kind of older, kind of had some side effects compared to the newer drugs, kind of had some side effects. Nothing as bad as having lung cancer, though. And this mouse, you can see the cancer's gone. It's melted away. It's exciting. Why is this one amazing? Because we've done this in a bunch of stories here. Why am I happy with this one? Because this example where we went from computer prediction to showing it works in cancer cells in a dish, which I'm not going to show you. We showed it works in this mouse, and about 18 months after the computer prediction, we got ethics board approval, and we've got patients recruited at Stanford already on this drug. We already have patients in a clinical trial. Now, if you've heard, this is supposed to take, for a new drug, $1 billion, $4 billion, 10 years. And what's exciting is if we can find new uses like this for existing drugs, maybe we can get them to the clinic much, much faster. This may still fail in the clinic, I don't know, but it might work. And we need more ideas like this. Small cell lung cancer has a 5% survival in 5 years, and it is not one of the cancers that has a targeted therapeutic for it yet. There are companies working on it, but there's nothing out there like we're used to seeing for the other cancers. So maybe this could do some good. We'll see. And what's amazing, too, is we start to learn the Stanford way. You got a platform working, you can even spin out companies on this. So we spun out a company called Numeti because we realized it's not just one pharmaceutical company, but all the pharmaceutical companies are having trouble filling their pipelines of drugs to sell. Their internal discovery isn't working well. Why don't we figure out new uses for the existing drugs and try to get those to market faster? So we spun out Numeti, and already Numeti had a pharmaceutical deal, and they got their funding and all of that here, too. So something in my lab to spin out companies now. Now, the other source of big data out there is really in your genome project. You heard a little bit about DNA and the whole genome project and how it was finished in 2003. The genome project cost $3 billion and was completed in centers like this one. This is a picture of the inside of the Whitehead Institute. You can see all the different robots and people staffing this and all the different facilities out there. And today, almost 10, 12 years later after the completion of the genome project, we're talking about getting your genome with devices like this, a USB stick. Right? So instead of this 13 years, we can, using technology from Illumina, we can get your genome in 27 hours. It's down to about $1,500 instead of $3 billion. And eventually, we'll even have it as a USB peripheral just to get your DNA. It's kind of an amazing world. What we learned right away, though, was the data. Getting this data is not the hard part. The hard part is interpreting all of these sequences. And what do we do with all of these sequences and the data? So we start to realize another source of big data that's out there to help us interpret this data is all the publication out there, all these publications. Where are all the spots in the genome that actually have something to do with diseases? And it turns out we can use text data, reading papers, even manually reading papers, to interpret this digital data. So we started reading 1 paper, 50 paper, 100 paper. Here are 1,000 papers. We've actually read more than 15,000 papers because you have to correlate and curate all that data to help us interpret the genomes. And what we've been able to do then is working with our first patient, Steve Quake, and now many others, we can start to make predictions. What diseases is someone likely to get in their lifetime? Here you can see just a bunch of different diseases, obesity and depression and diabetes at the top there. The black arrows were what we thought the risk would be, just given statistics. And the red lines and the blue lines indicate increase and decreased risk for this individual, Steve Quake, based on the genome. And if we can do this now for one person, we've been now seeing that we can do this for thousands of people. So what you're seeing on this graphic here is something that just came out, and we mashed this up with Google Maps. We could start to really study where did we get as a human species? Why do we even have spots in our genome that give us diseases? Wouldn't have those been selected out? We start to look at global populations now. This is not just going to be a US-only thing here, right? Genome projects. Already a lot of sequencing happening in China, Japan, some in India, a lot in Africa now. This is a worldwide phenomenon to figure out why do we have these diseases around the world. But actually, the most exciting thing for me in DNA is this kid. If you haven't heard of this kid, Nick Volker, this isn't work at Stanford, this is done in Milwaukee. This is a Pulitzer Prize winning story about this child who spent the first 5 years of his life in the intensive care unit because he continued to have bowel problems. Again, thinking about inflammatory bowel disease. No one can figure out what's going on with this kid. He goes in for surgery after surgery, and finally they sequence him, they sequence his parents, realize he has a new mutation. That led the treating doctors to think about doing a bone marrow transplantation in this kid, a stem cell transplantation, and now this kid actually goes to school, and he's supposedly going to have a healthy life now, moving forward. This is the first child saved with DNA sequencing. I think we're going to have many others like this. These stories are almost brand new. I'm only 2 or 3 years old now at this point. This is the revolution we're talking about. Big data also comes from other smaller devices. It's not just about the molecules. We all have been talking about Fitbits and Withings and all these personal monitors and quantified self. You know, it's funny. I used to kind of chuckle at these folks, these quantified self folks, until I started wearing a Fitbit. And on the bottom there, you see my own BMI graph. So I'm putting it out there for you, okay? You can see I came to Stanford, life got comfortable, I started to gain weight like crazy, and my BMI was in the red zone there. In January, I started to wear a Fitbit. So I keep track of what I'm eating, and you can see my weight has just plummeted here. I have a long way to go to get to green, but man, there's no way I would have been doing this if I didn't start counting and just measuring these things here. Data also comes from the policy side and the financial side. I was in Miami, actually, the morning this broke in the Washington Post, where University of Miami, it turns out, charges 3 times more than the neighboring hospital across the street, Jackson Memorial. If hospital policy folks and analysts don't stay on the top of this data, the reporters will get to it in this area of sunshine and letting this data out. So that's the policy side and cost side. And I think the next big thing that I'm investing my folks and my staff on in lab is on the clinical trial side. It is an absolute tragedy that half the clinical trials in the United States fail. Half the trials around the world fail. That's not the tragedy part. The tragedy part is we don't publish papers on the failures. They're not acceptable. And forget about the papers, we never see the data. And we're entering this world now, we're running this through the immunologists, through a part of NIH called NIAID, Allergy and Infectious Disease. That transplant data and infectious disease data, we don't care if the trial worked or not. People should get access to the raw clinical trials data. De-identified, of course, we don't want to know who these people are, but maybe someone else can come and figure out which patients is this drug working in and which one maybe is it failing in. And on the back there, you can see we let all this stuff out in MySQL format, tab delimited format. People should be able to build apps on top of the trial data. Why do we wait for a physician to write a paper about it and then read a paper about it? Just go directly into an electronic health record in some ways. And that's just the beginning. With data on the genomes and pharmacogenomics and chemicals and the electronic health records, all this data at least available, more and more of it becoming publicly available as well. It's just the beginning of this field. So to close this out, I mean, what do I think about big data in biomedicine? What is this all about? This organic data, I guess is what we call this. It's about algorithms, it's about programmers, it's about Hadoop and cloud and databases and high-performance computers and mobile. I actually don't think it's about any of these things. Big data in biomedicine, to me, is about predicting the disease before it strikes, explaining the rare disease that defies experts. Big data is finding drugs for diseases lacking attention. It's making sure we do the right and safe thing for patients. And it's also an amazing platform for biomedical innovation and entrepreneurship. Big data is out there. But I can sum all this up with one word. Big data in biomedicine is all about hope. It's hope for physicians, for patients, for providers, for families. The data's out there. It's up to you and us to do something about it. Can't do any of this work without a long list of collaborators and support. A big support now comes from this Big Data and Biomedicine Conference. I've got to put in a plug for that in May. It'll be in this room. I have a long list of collaborators, too short of time to even mention them all. And I, of course, have to thank endowment support from the Lucille Packard Foundation for Children's Health, the National Institutes of Health, which is paid for by your tax dollars. Significant chunk of it closing down on Tuesday because we just can't figure out the funding. March of Dimes, HHMI, Hewlett Packard, and a large amount of support. I have to always thank my daughter, who gives me inspiration to work on this work, Kimayani at the bottom there. And I always thank my wife, a molecular biologist, biochemist, who now starts companies with me and lets me go early Sunday morning to give talks like this. Thank you very much.